Okay, so the main difference between adaptive immunity and then the non-specific one is that adaptive immunity is able to defend, the body is able to defend itself against specific invading agents. So specific bacteria, viruses, what have you. So again, there's this idea of specificity, and then the other part is that it also has a memory. So the first time you might get sick with whatever it might be, but then the second time you may not even know you were infected to begin with, because you don't even develop symptoms. Our main two cells are going to be cytotoxic T cells. We'll also talk about helper T cells. So I guess T cells and then B cells. In general, if the antigen is an infected cell, like it's already gone into a cell and it's infected it, it's going to be the job of the cytotoxic Ts to kill it. If the antigen is still in the bloodstream, meaning it hasn't infected a cell yet, then it's going to be the job of the B cells in their active form, which are plasma cells, to produce lots and lots and lots of antibodies and basically to trap the antigen before it can ever infect a cell in the first place. And really these two are going to work together along with helper T cells to help protect us from getting really, really sick or dying. Okay, so here's again just a brief overview before we get into more specifics. There's two types of T cells, as I mentioned, helper T cells. These are going to activate or enhance the activity of the cytotoxic Ts and plasma cells, and then those cytotoxic T cells, which are going to actually kill infected cells. Now, if you remember back to when we talked about the thymus, these T cells are also going to need the help of something called an antigen-presenting cell because they don't recognize it. If they see it, they have to have it presented to them. Whereas B cells will become plasma cells when they themselves recognize the antigen. They don't have to have it specially presented to them. And it's in this active form as a plasma cell that B cells produce antibodies. Okay. But both B and T will have to recognize that there is a foreign microbe tissue, whether it's presented to them or not. Recognize the difference between a foreign thing and yourself, right? Because you don't want to attack healthy tissue. That's how we have autoimmune diseases, is when that happens, when those T cells kind of get a little confused. So they have to be able to differentiate between you and not you, and then attack the thing that is not you. So how do we recognize microbes, or how do these cells recognize microbes? Well, a moment ago, remember we saw that slide with the natural killer cell and the MHC1? Let me scroll back up to that briefly. So remember here we had our natural killer cell, and again, here's our normal cell. It had this MHC1 on its surface, and that inhibited the natural killer cell from killing it, and this target cell didn't quite have that, and so it killed it. Well, it's going to be like that. So basically, the MHC1 and MHC2, what that MHC stands for is a major histocompatibility complex. See, a lot easier just to say MHC. These are transmembrane glycoproteins. It simply means that this protein has some sugar molecules on it, and it goes across the cell membrane. You have unique MHC antigens. This is, again, why transplanted tissue becomes rejected. So if you have an MHC that is going to be different than someone else's, your body will want to reject that tissue. So we saw just a moment ago in that natural killer cell slide was that the body cell had an MHC1 molecule, so that's built into some cell membrane of all cells except red blood cells. This is why we can donate red blood cells to one another. That's why you can have blood transfusions. All we have to worry about there are the A, B, and the Rh antigens on those surface. We don't have to worry about the MHC antigens as well, thankfully. Every other donated tissue you do, and that's you've heard of people trying to find a good match for an organ transplant. What they're talking about is trying to find someone that has a very, very similar MHC1 molecule, because the more similar it is to yours, the less likely it is that your immune system will reject it. Okay, so MHC1 are built into cell membranes of all cells except red blood cells, and they are unique to you, right? Unless you have an identical twin. MHC2 is only on the surface of something called an antigen presenting cell. Remember a little while ago I mentioned to you that T cells need to have the antigens presented to them. They don't recognize them all by themselves. Well, the cells that do that presenting are called antigen presenting cells. It's very simple. We already talked about dendritic cells when we talked about the thymus. B cells can also be antigen presenting cells. And then most macrophages will also be antigen presenting cells. All right, so just a quick thing on antigens. Just remember we talked about these already in our blood types as the A antigen, B antigen, Rh antigen, right? So it's simply a substance that's recognized as, in this case, as foreign and provokes an immune response. In the case of your red blood cells, those antigens would provoke an immune response in somebody else if they didn't have a compatible blood type. You can further refine antigens into what are known as epitopes. So this is a specific part of the antigen that is recognized and bound by the immune system. And here's the deal. Every B and every T cell has its own unique set of transmembrane proteins that will have its own unique receptor in the cell membrane. So every T cell, so let me try to say this again. A specific T cell will have a specific receptor that will only recognize a very specific epitope of an antigen. So that's why, if there's a slight variation in the virus, a vaccine may not work. So we have to get different flu vaccines every year. Depending on whether or not you're on the East Coast or West Coast, you may get a slightly different vaccine because if the epitope is just different enough, your immune system may not recognize it. Okay, hopefully that makes sense to you. Again, so these cells are going to recognize a very specific part, and each T cell and each B cell recognizes something different. Okay, so it's not like all of them recognize a variety. They recognize one specific thing. Another one might, so if an antigen presenting cell brings a thing up to a T cell, if it doesn't have that specific receptor for that epitope, it doesn't recognize it. So then the antigen presenting cell will go to a different T cell and goes, do you recognize it? That one goes, no, I don't recognize it either. Okay, so then the antigen presenting cell goes to another one, and so on and so forth, until finally one of those T cells recognizes that epitope, and then it will respond to that. These antigens can be anywhere from microbes to toxins to pollen, again, transplanted organs, incompatible blood cells as mentioned, so on and so forth. In order for something to be considered an antigen, you have to be able to provoke the immune response. If it doesn't provoke the immune response, it's not an antigen. So you can have things like titanium in our bodies and it doesn't provoke an immune response. And then it also has to be reactive. In other words, it has to be able to react to cells or antibodies it caused to be formed. And as I mentioned, we have self-antigens, those MHC1s that say, no, this is me, do not attack me. Okay, so let's just say that we have an antigen. We are going to have to process that. So if you have this whole big microbe that is invaded, you're not going to just take that whole big microbe and present it. You're going to break that down or your cells of your body are going to break that down into very tiny little parts and then present those little individual pieces and see if something recognizes them. So the general process is an antigen, the proteins are broken down, they are associated in the MHC molecule. The antigen MHC molecule is inserted into plasma membrane so that where other cells can see it. The T cell that has the compatible shape will bind, and that will trigger a response. If a T cell sees that it's just self, it's just you, like that natural killer cell saw that it was just a normal cell, it leaves it alone, right? If it is foreign, it doesn't recognize it, it sees it as not you, then an immune response will occur. And either the antigens are processed either via an exogenous pathway or an endogenous pathway. So we'll talk about those two. So an exogenous antigen, this will be associated with an MHC2 complex. So these are antigens that are present in fluids outside of body cells. So exogenous, just think exit like they're outside of the body cells. So they haven't invaded the body cells yet. So if they haven't invaded the body cell yet, this means that an antigen-presenting cell has to come along, find it, and process it to present to a T cell. Okay. Usually these are in areas where you're most likely to come in contact with foreign materials, so epidermis, the dermis, mucous membranes, lymph nodes. That's where we see predominantly this exogenous processing occurring. That's also where these dendritic cells and macrophages and B cells are, right? They're in that lymphatic tissue. Here's just an image of that. If this is too complicated, then you don't necessarily have to worry about it. So here you have this DC is a dendritic cell, and what you're seeing here is that it's basically processed an antigen, and it's putting it into the cell membrane. Here's a B cell. It's also recognizing the antigen. It's going to bind that, cut it up, and present it. That's all that this is trying to show you. And then here is a T cell, and it's going to either recognize it or not. That's all this is trying to show you. Okay, so again, that was exogenous antigen, so something that hasn't invaded a body cell yet. And ever so often, your immune system doesn't get there in time, and microbes invade a body cell. Now, the big thing is you're going to have to kill that body cell, because what you don't want is them dividing inside the cell, producing thousands and thousands and thousands of these microbes or viruses, right? So most cells that are infected can process and present these endogenous antigens. So basically, instead of that cell going, "It's me, don't kill me," it takes away the thing that says that it's you, it's itself, and puts in its place a little piece of the antigen. So even though like that antigen is dividing rapidly inside of it, it can't kill off all of it. It can kill some of it and go, "Hey, please kill me. I'm infected," right? "I'm gonna turn into a zombie. Kill me now." And that's basically what you're seeing here. So there's a virus. The cell digests some of that and presents it so that the cytotoxic T cell can identify it and sees, "Oh wait, that's not you," kill it, right? In the case of the MHC2, which is again the exogenous, we're going to have to take in that microbe, break it down, and present it. Okay, so hopefully all of that makes sense. So really this is like the first step. We've had an infection. These are the different ways we can present these things, either on a cell that has been infected via an MHC1. So again, if I'm an infected cell, I am going to put out on my surface a marker that says, "I am infected, kill me." That is an MHC1 molecule. If I am a macrophage, a dendritic cell, or a B cell, only those three, and I recognize and I gobble up a foreign microbe, I'm going to present that in an MHC2 molecule. Okay, so, and then I will present that to the T cells saying, "Hey guys, there is this thing out there. Go look, see if any cell is infected, and if it is, you'll know because it's on the MHC1, go ahead and kill it." And again, remember, only a very small subset of T cells or B cells will recognize the epitope for a specific antigen. Okay, if you have any questions, please let me know.